Thanks for inviting me to fill you in on things. Thanks to everybody for coming to hear about this. I should say at the outset, acknowledgement, much of this is a promissory note. We're about 1 year into using these devices now. We've made some accomplishments, I think, on the sort of use of the devices in general for our purposes of trying to gain access to lots of neurons, to very large numbers of neurons from which we think we can surface an enormous amount of information, just as introduced in the beginning. One of the major challenges here is to sort of move up to the level of, instead of recording from single neurons and identifying simple sort of trigger feature and receptive field properties, to identifying what large populations of neurons are doing, and see if we can see emergent properties come out of looking at populations of neurons instead of neurons one at a time, and asking how they sum up in a simple kind of arithmetic. So our topic of interest as introduced here is really kind of the high level of function, of cognitive functions. I'm interested in things like cognitive maps. Now, you've probably heard a lot about cognitive maps recently with respect to the hippocampus. I don't mean the ones that are restricted specifically to maps of geographic space. What Edward Tolman meant by cognitive maps when he introduced the term earlier was really a map of cognition. In a sense, a map of life. How do we put together all of our memories to come up with a structure of knowledge that we have? That's a pretty ambitious goal. We think we can make some headway on that if we can record from large numbers of neurons instead of neurons one at a time. So we've begun to use the Inscopics technology to head in that direction. What I'm going to tell you about today is some sort of preliminary day of that sort that tries to answer sort of one of the deeper questions that even when the challenge was framed, that if we recorded from loads and loads of neurons, we would find out an awful lot more. The sort of unanswered question so far is, how would you do that? What would you do with all those neurons if you had them? I've heard that question asked many, many times. What would you do that you wouldn't do with just neurons one at a time, for one after another after another? What would you get by recording simultaneously from lots of neurons? And I'm going to tell you about some progress on that front that really goes back to the old technology, in this case tetrode recordings, that delivers that sort of promissory note. And in a sense, I'm suggesting one way to approach answering that question of what would you do with all those neurons. And then I'll tell you a little bit about our progress with using the Inscopics technology to get all those neurons to answer the questions we want to go after. So let me start out by pointing out that even though an enormous amount of background, enormous amount of what effectively when I grew up, and I'm one of these old dogs who's effectively trying to learn new tricks now, but the old dog view of things, which really came out of a terrific exemplar of which was sort of the Hubel and Wiesel view of visual cortex, for example, is every neuron has a trigger feature and a receptive field, and essentially encodes one dimension of experience. And that aspect, that kind of thinking has been applied to the hippocampus as well, in encoding specific places in space or head direction or other unidimensional parameters. But actually, if you let animals move freely and do the kind of complicated things we want to understand, to learn and remember and explore their environments, neurons typically do an awful lot more than that. And this was brought to the forefront recently, a few years ago now, by Stefano Fusi, who analyzed some data from Earl Miller's laboratory in prefrontal cortex, which is simply exemplary of the higher order cortical areas, which include the hippocampus too, that neurons in these areas actually don't have single unidimensional feature coding properties. They're complicated. They have very complex properties. They have what's often referred to as mixed selectivities. They do a whole bunch of different things. They're responsive to multiple dimensions at the same time. It's what you end up with, in a sense, the direction, this sort of frustrating direction this could be headed in, is to say every neuron sort of encodes everything to differing degrees. And how are we ever going to make sense of that? Well, there are some approaches emerging that might allow us to do so. So I'm going to take a few minutes to kind of introduce the approach and some of its success at even a level below the hippocampus. And then I'll tell you more about what we're finding out about the hippocampus itself. One of the approaches, and this is just really one of many, but one we found very intriguing and potentially successful so far, is something generically called Representational Similarity Analysis. In short form, what it amounts to is simply asking of a very large population of cells, how similar is the response pattern across a large population of cells under multiple different conditions, and measuring those similarities, ultimately constructing a structure of how they work together. I know that's awfully vague, but I'll get very specific as we go along in this. Let me start with an example of how it's been applied. And in fact, mostly it's been applied actually so far in functional imaging data to really scale up to loads and loads of neurons, although not one at a time. So in functional imaging data, what some experiments do when looking in particular, in this case at the ventral temporal cortex and visual responses, here's an experiment that comes out of Haxby's laboratory a few years ago now, where they simply show the images of those different species of animals to a person looking at a screen, right, and took the multivoxel pattern that you see in this visual cortical area and compared that pattern, that simply cross-correlated the pattern of voxels, the multivoxel pattern, across the different kind of images they were looking at. So that's the first step in the process. So for example, one can see, in this case, the two insects. If you look at the cross-correlation between the two insect visual things have low dissimilarity or simply high similarity. So the voxel patterns were very similar for two insects, right? And they're very similar actually for two birds. So you can see here, right? There would be the similarity and high similarity for two primates, okay? And low similarity, that is greater dissimilarity, for example, for an insect versus a primate, just to give you a flow of how this goes. So what do you do with all of this kind of data? What you get is all these different similarities and lack of similarities. Second stage of this process is to apply a sort of dendrogram analysis in this case. And essentially what the dendrogram analysis does is look at all the possible events, that is the different animals in this case, right, and ask pairwise which two produce the greatest similarity to each other. Well, as I said before, essentially the two primates produce the greatest similarity, the two birds, right, and the two insects. So one ends up with clusters that correspond to natural categories that we perceptually and semantically know about, primates, birds, and insects. So now you simply go iteratively through that process again. In this small group of exemplars, one simply then finds that these two of the three clusters here, this one, this one, and this one, the two clusters together, not surprisingly, are going to be the vertebrates, and those are separated off from the insects. So sounds kind of simple. It sort of better turn out this way, and one would think the brain would categorize phylogeny essentially the way that we do, although I still find it kind of amazing that here we have physiology sort of recapitulating phylogeny, right? Phylogeny pops out of the system. So here's an organization of percepts and concepts of different species that one can identify, can actually identify the organization of that information. And one can do the same thing at a single cell level. I won't go through all the details of this one, but these are data from Tanaka's laboratory where they recorded from lots of single neurons in the infratemporal cortex, right, and simply pooled all the data from lots of neurons and coded one at a time, having shown them all these different images. One can go through a simpler representational similarity analysis of looking for similarity of clusters and applying the clustering algorithm to pull out something that's sort of partially phylogenetics and partially sort of body parts or animate and inanimate parts, and again, create a structure of knowledge, in this case of a sort of perceptual space, right? So that all sounds, again, kind of straightforward, a proof of principle. We'd hope the brain would work, we'd hope these visual areas would operate this way, to separate the categories which we from psychology know operate in this fashion. So the question where we wanted to pursue it was to take it further into a structure for which it's not so obvious what the organization should be, into the hippocampus. So in the hippocampus, what we think is going on in the hippocampus is the hippocampus makes these maps, these cognitive maps, sometimes called schemas, of memories, essentially, that creates an organization of memories that parallels in temporal cortex would be an organization of perceptual space. So just as we have perceptual spaces in the temporal cortex, we think in the hippocampus one has an organization of memories. It's not so obvious in advance how the hippocampus would organize memories, but that's what we're after. And we think the same methodology can be pursued to find out the organization of memories within the hippocampus itself. So just to show you the behavioral task, the paradigm we use to make some progress on this front, here's a game that we have, in this case, rats playing. Mice play the same game. I'll show you a little video of it later on as we're starting to collect data from mice. In the beginning here, these are going to be rats playing the game and recordings from tetrode recordings from lots of cells at once, although nothing like what you can get from the enscopic system. So we're going to be talking about 30 to 50 neurons at a time, as opposed to later on I'll tell you about much larger numbers than one can get with the system. So basically the way this game works, just to sort of bring you into the lab and the picture about it now, is the rat walks back and forth between these two boxes, these two environments, or what we're going to call two spatial contexts. He walks into one of these contexts, let's say in the left one here in context one, and he's presented with two objects that could be in either spatial position. So objects A and B here could be in either of these up-down positions here. And the rule of this game is that over here in context one, item A, the yellow one, will have a reward buried in it, and item B will not. So all he has to do is a simple discrimination of picking A and not B when you're in context one. Very simple. Rats can learn that sort of thing in nothing flat. It's a little bit tougher when, in addition to that, whenever he enters context two, he sees the same two objects, which again could be in either of these two spatial positions, but now the rules are opposite. So over here in context two, it's object B, the purple one, that has the reward, and not object A, the yellow one. So he has to use the context to identify which of the two objects to pick. He effectively has to learn what events happen in what places in the world. And he has to learn about all 4 objects that could appear in any of 2 positions in either of the 2 contexts. So he's accumulating a good number of memories. In this case, effectively, there are 8 different memories, two different objects that can appear in any of 4 different positions. So how do we find out how these memories are structured together? What's the memory space? What's the cognitive map of all these memories? So to do that, we basically start looking at hippocampal neurons, and we're going to scale up the task as well. Now, just to show you about mixed selectivity in hippocampal neurons, let me just show you the firing patterns of a single CA1 pyramidal cell in an animal performing this task. So here, effectively, the 8 different memories he could form, item A versus B, in either of the 2 positions within one context, and all those same memories in the other context. And effectively, if one just looks at the period between these little arrows here, that's when the animal is sniffing at the stimulus here, what you can see is this cell fires rather robustly when the animal is sampling item A in position 1, much less so if he was sampling item B, so it seems to be a cell that likes item A. But actually it doesn't like item A so much if you put it in position 2, which would be the lower part down here, right? Much better in position 1 than position 2. Right? And over in the other context, it doesn't fire to item A anywhere, or to either of the positions anywhere. So here's a cell who has mixed selectivity. He's a gold standard for this sort of thing. This cell is active only if you have just the right item in the right position and in the right context, all 3 things. So how do we find out? And we have lots of cells. As we record these cells, we find enormous numbers of cells have some combination of these different parameters. They care about the object, they care about the position, or they care about the context, or in any kind of mixture and combination of those. And one is just kind of left with this pile of cells that encode all of the dimensions that are critical to the task. How can we sort out how these different responses, what these different responses tell us about memory and how it's organized in the hippocampus? Well, we do so by scaling up our endeavor in 2 ways. First, we scale up the behavioral task. So here's the task as I showed it to you in the last slide. The first thing we do is actually scale it up to have 2 more objects. So some trials are like the kinds of trials that can occur up here. Other trials are essentially the same, except now the animal's given a choice between object C and object D in either of the 2 compartments, following essentially the same rules. All right? So now the animal develops not just 8 memories, but there are really 16 different memories, 4 different objects that can occur in any of 2 positions, in any of 2 contexts, an awful lot of memories that kind of could potentially interact with one another. And how does he develop a sort of knowledge space or a cognitive map of all of these memories together? How can we join them all? So we come back then to our version of representational similarity analysis. Now I'll just bury you in the details of how it works very briefly and very, very superficially. So here would be the recordings from a small, very small ensemble of neurons, in this case 26 neurons that were recorded and identified simultaneously. We normalize their firing rates from the cells with the highest firing rate to the lowest firing rate. And essentially all we have here are the responses in terms of a firing rate over a little snippet of time, a little 1-second period when the animal is actually sampling one of these stimuli on just one of those trials. So here's a trial when he was presented with object A that was rewarded in this position and in this context, and here's some cells firing from those that fired at the highest rate to the sort of average rate and ultimately to the lowest rate. So that's the population vector response, not a single unit response, but the population vector response to that particular combination of all of the different dimensions that composes effectively one memory. So what do we do with that? Well, we compare it to what happens on other memories or on other trials. So in this case, to take the simplest example, we'll just take a repetition of the same trial that happened later in the same session. Right, so it's exactly the same parameters, item A, in the same position and context. And here's the population vector one gets from that. And if you just simply compare across these 2, you can see the patterns are very similar to one another. You perform a very straightforward correlation between these 2 population vectors. Is going to be a high correlation. Right? Not surprisingly, repeated events have repeated population activity patterns. That better be the case. If it weren't, we'd say the hippocampus doesn't care about what's going on here. It'd just be noise. All right, well, that's the first starting point, the simplest starting point. That better be true. But then what happens if we change one of those dimensions? So in this case, we simply switch to item C, object C, which is functionally equivalent to A. That is, it's rewarded for just as A is in the same position and in the same context. And now look at the population vector here. Clearly, this is more different than this one was to this guy, right? The population vector has changed a bit, right? The correlation is going to go down, which simply means as you change the object, the hippocampus is encoding the object property itself, and the comparison of population vectors tell us that object identity pulls things apart in representational space. They're farther apart, they're less correlated. All right, just as one more example, let's change something else. So here we'll stick with item A, but we'll put it in the other context. All right, which means that obviously it's in a different position, and moreover, it also has the opposite reward value in the the other context. Remember, if it's rewarded in one context, it's not rewarded in the other one. So all of the parameters essentially have changed except object coding. Now one sees a vastly different population vector, right? So what used to be really high firing rates up here are now lowest firing rates. What used to be the lower ones are mostly the highest ones here. These two are not only very far apart in representational space, they're anti-correlated with one another. It's as if the cells that are on under one condition are the ones that are off in the other condition, and vice versa. So one gets these various distances in representational space by comparing across all the possible memories, all 16 of those memories that can occur. Each one repeats on many different trials. So there are lots and lots of these pairwise population vector correlations that can be performed, and if you do them all, you get something like this. So now it gets a little mind-blowing to try to study it in detail. I'll just talk louder and louder, okay? So this is essentially a correlation matrix showing all the correlations between all the possible pairwise comparisons of every trial of hundreds of trials that occur in a single recording session. So essentially what's lined up here is all the possible events, or all the memories one can imagine. So here's item A that was rewarded in position 1 and context 1, and so on down the line for all the possible events that occur, right? Against essentially all those same events down here, so we're simply cross-correlating them. The diagonal is an event cross-correlated with itself, so obviously it's a perfect correlation, right? And the others are correlations to either the same kind of event or other variations in the events. And I won't try to go through the whole matrix here, but this is just the data from one animal in one session, but I'll highlight a few comparisons that you can see right away. Now, the high correlations are in dark red, zero correlation would be in white, and blue shows negative correlations. So here what you can see essentially is that functionally equivalent items, A and C, always mean the same thing in the same place, are fairly highly correlated, right, if they occur in the same location and in the same context. If you simply move the same two objects to a different position within the same context, you can see the correlation goes down, less red here than was red here. But they're still largely positively correlated. They're still largely red. All right? So again, pulling things apart in representational space, if you move, in this case, across space within a context. But now what happens if you move across contexts? Well, all comparisons across contexts are basically all in this big square down here. And what you see here is a sea of blue, which simply means everything that happens in one context is virtually anti-correlated with everything in the other one. So this is a highly systematic representation that responds to all these changes in powerful ways of pulling up different populations of the neurons. But essentially, if you could stare at this all day and drive yourself crazy looking at all these, and at this point it's still telling you about a lot of correlations between populations. It doesn't tell you yet about how the different memories are organized in the hippocampus. But here's where it comes from if we move to the dendrogram phase, if we now try to put this together in an organization. So the way this works, just as it did in looking at ventral, temporal cortex, and perceptual space, was to align all the possible events that occur. In this case, all those events are aligned on the bottom here. So this is A in position 1 and context 1 and so on down the line for all 16 events. And again, by virtue of the way this analysis works is simply pairwise, say which pairs of all 16 events are closest together, are most highly correlated. And the answer is this. Turns out to be highly systematic. So the events that are most highly correlated are, for example, two functionally equivalent items, A and C, within one position and within one context. Here's another one, B and D are also functionally equivalent. They mean the same thing in one position and in one context, and so on down the way. So the lowest level of system, the events that are closest together in representational space are two different events, A versus C, for example, but that are happening in the same place and in the same context. So now we go through this process iteratively. One simply asks, and here we have 8 clusters. What if we pair those up? Which pairs are closest together? And again, one gets a systematic outcome by which events that have different meaning in the same position in the same context are tied to one another. So things are separated or associated by the outcome, by what they mean. All right, cutting across them. And to take this one step further then, now we have 4 clusters. Which pairs of those are closest together? Well, it's the things that happen at different positions within a given context. So here we have everything in position 1 is here, everything in position 2 is here, and those guys are connected together. And the same thing on the other side, in the other context, okay? And then finally, of course, there's only one link left. But it's worth noting in the case this top link here has a negative correlation associated with it. That was all those anti-correlations. So just running through this once more, I'm sure this is foreign to most people who stare at these complex graphs here. Effectively, what this is saying is the first thing the hippocampus does is say everything that happens in this context is going to have a completely different representation or largely different representation than what happens in the other one. You want to, in this the case of this task, since everything that means one thing here means the opposite over here. Whatever's good here is bad here, and vice versa, right? Then what first thing you want to do is suppress all the memories over there when you're over here, and you want to pull up all the memories here. So the hippocampus seems to do that at the outset. And then within each context base, that is within one context, all right, now we want to associate things together in a systematic sort of way by which things are first separated or by the positions where they occur. Did it happen here or did it happen there? Then it says, okay, is this good news here or bad news here in that position? And finally it says, okay, which object was it? So it has all the details down. So the hippocampus really encodes everything. Certainly it doesn't, unlike what you may hear from others who like to talk about the hippocampus. It's not just up there in the spatial plane of what's happening, although those are very prominent parameters here. It's mapping these events out in space, but it's also mapping what happens in different places according to the two properties of exactly what perceptually happened, which object was it, and what does it mean? Is this good news or bad news? And the hippocampus cares about and systematically represents all of this. So I like to think of this as a cognitive map of these memories, or a kind of memory space that pairs them together. And it's something that emerges out of the analysis. This organization can't be seen in individual neurons. And once again, remember, individual neurons simply code for all these different dimensions. And that's it. You sort of stop there. This tells you how they're organized. One can go further with this. There's some other fun tricks you can do with it. You can look at the dynamics of the representation. So actually, instead of picking out data for a whole second of data sampling, a period like this long in here, and just getting a single data point for how a single instant, I should say, a snapshot of the organization, one can look at the organization moving over time. So this is looking at those same organizations, picking out the same parameters, over short snippets of a couple hundred milliseconds. And what you see here is essentially that all those dimensions that are mapped across the different memories actually operate in sequence. So the first thing that gets encoded is actually the position the animal goes to. Not surprisingly, he's walking into one of the corners. So the first thing that gets encoded is, which corner am I going into? And then right after that, when he gets there, so this is when he gets to the object, one gets a rising coding of, okay, which object is it that I'm sitting here staring at? And then finally, what happens at the end of that succession, succeeding it afterwards, okay, what does this mean? Is this good news or bad news? So one can actually look at shorter and shorter time windows. Now, the magnitude of predictability here is strong enough, certainly, to be highly significant, but it can get much stronger. We can look at short periods of time and look at the evolution of this if we only had more and more neurons. So if we could move from population vectors of 25 or 50 neurons to population vectors of hundreds of neurons, one could get into the resolution of this in much greater detail. There's one more thing one can do with this and actually look at the dynamics of this over larger chunks of time across days and how information is actually combined. And again, even at this simple level of small numbers of cells, one can begin to see an inkling of that. In this case, what we did was we have to look back in time. So the data I've been showing you so far is sort of way out here in training when the animal is really expert at the task and getting all the possible combinations of stimuli. But if you look earlier in training, back up here, the animal in the beginning of training really just gets the A versus B problem. Remember how I described it. And then one day we shift over and train them on the C versus D problem, and then we only train them on the combination of those later on, and that was the data I presented so far. But if we move back in time to simply what's happening at the end of the AB problem, we collect a bunch of cells, they have their firing properties, and then the very next day we can hold on to many of the same cells, but that's another advantage of Inscopics. We can probably hold on to all of the same cells, or a very large fraction of the same cells across days. But even in recording in small numbers of the same cells, we can record the same cells when you suddenly confronted for the first time with these unfamiliar items C and D. And what happens? How fast do new items get incorporated into this map of memories? And the simplest way to look at that is another one of these correlation matrices. But in this case, we correlate, for example, what happens on two functionally equivalent items. So this is item A in position 1 in context 1 against C in position 1. These are two items that are functionally equivalent, and they have a very high correlation between them right off the bat. So essentially, as soon as you learn how A and B works, you can insert C and D almost immediately. Although in this case, again, our window of time is a whole session. So effectively, to think of it sort of idealistically, when the animal learns about A, B, this is the structure, this is the map of memories. And when you learn C, D, you simply fill in things very rapidly to accommodate the new items that are paired up here. And what's amazing about this from, again, the viewpoint of psychology and memory, so to speak, is that A and C have never actually been seen together. Right? Yet they're incorporated to be very close to one another. That allows one to basically fulfill the concept of functional equivalence. These two things are kind of the same thing, even though I've never actually seen them together. It's a kind of an amazing thing in how psychology of memory works, and it does work exactly that way, and the hippocampus, of course, knows about this. I want to take this kind of one step further to press your patience in listening to all this. We've taken it further now in recent days to beyond the map and the hippocampus itself to look upstream in two other structures that feed a lot of information into the hippocampus. So much of the dogma in the field, some of which I've promoted to a great extent, is that the lateral entorhinal area, which feeds a lot into the hippocampus, is essentially an object-specific area. It's the end of the classic what stream of cortical areas, right? And indeed, other studies have shown in the past that individual cells in animals performing simple recognition tasks typically respond selectively to different objects and sometimes to their familiarity as well. One never thinks of them as encoding anything about space, because they're part of the what stream, not part of the where stream. Conversely, there's the where stream, which ends up in the medial entorhinal area, which has a famous cell type we know as the grid cells, right? So that's a perfect map of space. So these another unidimensional coding neurons that simply encode what position you're in this spatially periodic fashion. And we think of these as two opposite streams that don't share anything with one another, right? Objects being encoded here, places being encoded here, and they finally only get combined in the hippocampus. So sounds good, sounds like a simple story. What if you actually put animals in a situation where they have to learn about objects and about spaces at the same time, in particular, learn where objects occur in space and what they mean? That, of course, is the task I've been telling you about already, where animals have to learn where objects mean different things in different places. If you do that and record from single cells, and just to show you how dramatic the dilemma can be between single cells and population coding. Here's the patterns, and I know they're kind of complicated, so I'll just give you a couple highlights of what happens. So here's a neuron in the lateral entorhinal cortex. It really prefers item A here to B, for example, and it does the same thing here. Likes A more than B, likes A more than B, but oops, doesn't like either A or B over here. So this guy has a spatial component. It's not supposed to have that. It's supposed to only care about the object, right? And we found loads of cells, just like hippocampal cells, that care about, to differing extents, about which object it is, where is it, which context it's in. All right, so that would be bad enough. People don't care that much about lateral entorhinal cortex. Here's the cells we care about that win the Nobel Prize for being spatially coding neurons. And this indeed is a grid cell. For those who know the hippocampus, you know this signature of a perfectly wonderful grid cell here. Well, put into this task, here's a guy who likes item A and not item B in this position in context one, and it doesn't care that much about other things in other places. This guy is really encoding, again, all the same mixed selectivity parameters when you release the animal to do more than simply wandering around space, which is what they do here. So these neurons in these systems are not very good feature detectors for individual unidimensional features like exact location or particular objects that when put in the real world doing real things, they exhibit these same kinds of high-level mixed selectivities. And in fact, if you simply do measurements of selectivity, which we've done on all these cells, it's pretty hard to tell the difference between lateral and medial entorhinal cortex. And for someone in this hippocampus field, that's kind of disappointing and frustrating. So these two areas are the same thing after all. Well, actually not. They can be rescued. We can rescue it. And the answer to rescue, of course, is, you get the push here, is representational similarity analysis. I won't take you through the details again, but just the upshot, the end point of this is if you look at the lateral entorhinal cortex here in the same analysis we applied to the hippocampus and for either lateral or medial entorhinal cortex, the first thing both structures do is, again, separate things by which context are you in. So the whole medial temporal lobe system first says everything that's good that I want to hear about over here, I want to suppress when I'm over here. All right? So you want to separate representations across these two meaningfully different contexts. But underneath that, the differences emerge. So lateral entorhinal cortex, once you figure out which context you're in, the first thing it does is separate which objects you're in. So essentially identify, okay, which object am I looking at now? And only secondarily does it care about where the object is. All right, so it's looking at an object, says, okay, that's object A now, okay, where is it? All right, that's a secondary property. Conversely, medial entorhinal cortex looks at positions, so it does show its spatial coding properties, and only within positions does it discriminate the objects. So the organization of the different memories, effectively the different events, is different. It isn't, both areas encode all the information, but you can pull out this emergent property of how is that information organized and how is it organized uniquely in each area. So this is where we really get excited about the use of very large ensembles of neurons where one can identify properties that can only be seen at the population level, whereas at the single cell level would be kind of frustratingly complicated. So where am I? I'm just going to lump these together now in this sort of short presentation to say I think there are really three unique advantages that are really qualitative advantages. So often when people talk about recording from very large number of neurons, it's almost just for efficiency. Wouldn't it be easier if I only need 1 or 2 animals and I can get 1,000 neurons out of that many animals? I'll just do experiments faster. Not sure that ever turns out to be true, but at least that would be the simplest promise. But that doesn't tell you any new information, right? But I think 3 kinds of new information can come out of this. One is not only can you find out what individual cells do, you can find out how the information is organized within a population of cells. You can never do that with individual neurons. Secondly, there are properties that emerge on populations that you can't see by just looking at collections of neurons that seem to do everything, right? And thirdly, you can actually look at the dynamics of the entire population if you have enough neurons. Now the efficiency comes in to do the population coding. So when you do these kinds of analyses, and there's several other parallel Bayesian analyses and other ones that gain enormous power by having lots of cells that are being recorded at once, bigger N to do all of the analyses for. And I think all 3 things come out of that. So having said all that, I want to get a little bit into the technology here. Let's see if I can get this thing started and show you. Is this a mouse doing exactly the task you saw already? So here's the 2 contexts. Here's a mouse with an Scopex camera on, running around playing the game. He's going to reject one of those as the bad one. He goes to the new one. He pushes it aside and gets his reward. So mice are very good at these games, it turns out, and we're very happy about that. So we're doing these recordings right now. We've been recording for a few weeks. Kind of ran into a little roadblock. I was hoping to show you some of the data from that, but certainly the cells responded all these times. We're hoping we'll start out by replicating the rat experiments. Again, same sort of thing. Rats, mice are really terrific at this stuff. The other thing I wanted to show you, which I won't quite start the video yet, let me just introduce it first. The other place where we've made progress is kind of the other challenge of moving into the hippocampus and these complex tasks. And the aim of getting as many cells as possible is you have to be able to isolate individual neurons. Now, and those who've done tetrode recordings or other kinds of conventional cellular recording, we all know about cluster cutting and these very difficult and yet imperfected means of separating individual neurons, which in the hippocampus in particular is a very big challenge because the neurons are very close to one another. Multiple ones tend to appear on the same electrodes, so separating them is tough. And actually in video, it's equally tough because you're looking through a chunk of layers where the cells are kind of mushed again on top of one another in a very closely packed scheme. And we need to be able to separate them to know what each one does individually and the subtle differences between them. But now we have the challenge of starting all over from scratch. It was tough enough to do it with waveforms you get in conventional recording. Now we have to do it optically. So how does one do it optically? And of course there are some methods available. But one of my postdocs, David Sullivan, his name is across the bottom here, has succeeded in doing this with a new method he calls TENASPIS. So this is the raw recording, but with a temporal first derivative. So it just pulls out the cells and makes them sharper. Now you're looking at the second stage he does, which is to look at contiguous pixels, so you can get these blobs or regions of interest, which look like the other videos we've seen before. But the third step is so different that it actually looks across time and across frames, identifies the center of mass of cells that seem to have the same center of masses. So it can pull out cells that are really right on top of one another. It's worth running again, maybe? Just it goes pretty quick. And then, so it kind of reverses the usual process in the conventional way of doing this, of actually just simply of surfacing the sharpness of potentials initially by pulling out this temporal first derivative. So you see brighter flashes and then it goes dark because the waveform is coming down sharply. So it pulls out the images differently. But now it's just doing it to the whole template. Now it's actually pulling out individual blobs, simply as contiguous pixels. That has to be the case. But then the really crucial feature we think has been quite successful is to look across time for essentially the same blobs to occur. So even overlapping ones are going to have slightly different centers of masses. And if you can pull out the separate centers of masses, you can separate cells even when they're not exactly on top of each other, but heavily on top of each other. And the outcome of this, which I can show that, is this so far. So here's our favorite little guy for which we were able to separate out 1,500 neurons at the same time. And that's a scaling up from, again, about 30 neurons at a time to 1,500 neurons at a time. And we think we can go a lot further with the analyses I've been talking about by doing this sort of thing. So I encourage you, actually, I think I had it, just to jump back here to go to his dynamic poster that's going to appear on Tuesday morning. Yeah, Tuesday morning. That shows you exactly, for anybody who's interested, that shows you this technique that he's developed for separating neurons. And I think it's going to be a boon to using the Scopex machines. So thanks for listening.